So welcome back. We have looked at how to identify a lead compound, and now once we have a lead compound, we need to further modify it so that we can make the compound more drug-like. Okay. So we will be looking later as to what are the ways in which the compound is taken forward once we identify a lead, and how clinical trials are conducted and so on. But before we get there, we need to have a good understanding of whether we have the best compound to take it forward, because clinical trials are very expensive, and therefore we need to be sure that we have the best compound. Okay. So traditionally we are trying to synthesize more compounds, and using a traditional structure activity relationship, we have some idea about what are the next steps forward. Today we will look at a quantitative way of studying this, and this is called quantitative structure activity relationship. So this is QSAR. So let's start from structure activity relationship. So here you have a number of possible analogues that can be synthesized. So if we don't have a good idea about how to go forward, we will look more or less from intuition as to maybe this part of the structure should be changed or the other part of the structure should be changed and so on. But if I add up these combinations, it will be nearly infinity, and clearly we don't have the resources to synthesize infinite number of compounds. So what we need is we need a rational method, and preferably a quantitative method for planning the next steps. And since synthesis is actually quite expensive to carry out, we would need to take help from quantitative methods. So here is the approach that we use. We use a QSAR approach, where we aim to identify and quantify physicochemical properties. So after all, a drug or a small molecule consists of various functional groups. It consists of various structures in the molecule which have some physicochemical properties. So let's say there is an enzyme which we are targeting, and there is an inhibitor that we have synthesized, and now the inhibitor has to bind to the enzyme and inhibit it. Okay. So all these binding we have already looked at is all, majority of them are non-covalent interactions, and those interactions are important for the binding properties. Okay. Now can we identify various physicochemical properties of this drug molecule or of this inhibitor, and see which of these properties, if I change any of these properties, what effect it has on activity. So that is the aim of doing typical structure activity relationship, but we will attempt to do it in a quantitative manner. Okay. So once we have a quantitative manner, so let's say you have a straight line or a curve, I now know that if I make a particular change to a particular physicochemical property of the molecule, then it is going to have this particular effect. Okay. So this will help with obtaining trends, and now once I have this trend, then I can in some predictable manner say that here are the next 10 compounds that you need to make. Okay. So that will help the synthetic chemist to synthesize new molecules. Now when we are doing these structure activity relationships, this QSAR, we may also find that there are some certain molecules which don't appear in these trends, but they are still active. So those molecules will also become important, because now there may be some other physicochemical property that may become important. So therefore together these are the reasons why people attempt to, or we look at quantitative structure activity relationship. So what are the conditions? We can't do QSAR on all types of compounds, but we can do it on a small subset of compounds, and the condition is that it must be structurally related. Okay. So if I have a bunch of inhibitors that I have studied, and I have identified a lead compound, and that now I use SAR, that is structure activity relationship, to identify or modify that lead compound, and then I have, let's say, 20, 30, 40 compounds in that series, and I study the inhibitory effect. Now these compounds that I am studying are a variant of the parent compound. Okay. So therefore they are structurally related. Okay. And since they are structurally related, it is quite likely that they will act at the same target and perhaps have the same mechanism of action. So QSAR can be reliably conducted on this series of compounds. Next, we should use the correct testing procedures that have been used. So if I have a purified enzyme, we have already looked at it in the past as to how to identify enzyme inhibitors. So we typically purify the enzyme and then screen for inhibition of the enzyme activity. So these correct testing procedures must be used on the purified enzyme with this series of compounds. Okay. And keep in mind that it is preferable to get data which is comparable with the enzyme under the same conditions. Okay. So we can then study how each compound binds to the active site. And now QSAR cannot be very easily used to derive good conclusions from in vivo data. So you may have a lot of in vivo data with a particular compound, because in vivo data is going to have more difficulties, as because you have to think about pharmacokinetics, you have to think about biodistribution, and all these properties are not very easy to predict, and therefore QSAR will largely fail when it comes to trying to explain in vivo data. So, the conditions under which QSAR works is that we should have a series of compounds which are structurally related, which have been examined by appropriate testing procedures, and they should be on a purified enzyme and not in a in vivo model. Okay. So, one of the first things that people look at when it comes to understanding how a series of compounds works is hydrophobicity. Okay. As you know, since the cell membrane is rich in lipids, the drug must have some hydrophobic character for it to cross membranes and act at the target. Okay. At the same time, we need aqueous solubility for improved bioavailability. So, the hydrophobic character of a molecule can be determined by what is known as a partition coefficient. Okay. Partition coefficient is nothing but the level to which a particular drug molecule partitions in two different solvents. Okay. So, during synthesis, for example, aqueous soluble impurities can be washed off using a separatory funnel, which many of you may have used. Similarly, what we are going to do is we are going to determine what is the partition between octanol and aqueous solution. Okay. So, P is nothing but the concentration of the drug in octanol divided by the concentration of the drug in aqueous solution. Okay. So, in a separate module, we will be looking at how to determine partition coefficient, and therefore, I will spare the experimental details over here. Now, if I have a mixture of octanol and water or buffer, hydrophobic compounds will preferentially partition into the organic layer, okay, whereas the hydrophilic compounds or the aqueous soluble compounds will preferentially partition into the aqueous layer. So, therefore, hydrophobic compounds will have higher concentration of the drug in octanol, and therefore, the P value becomes high, whereas in the case of a hydrophilic molecule, it will have low P value as a converse. Now, once I have a lead compound, I can vary the substituents and then study the effect of the change on the partition coefficient P. This effect is what we would want to study and understand whether it has any effect on the activity of the molecule. So, keep in mind, we are designing, let us say, an inhibitor for an enzyme, and so we want to study how hydrophobicity affects the activity. Okay. So, here is a case study. So, here they have taken about 42 compounds and they have studied the ability of this group of molecules to bind to serum albumin. Okay. So, they find that if I take the ability to bind serum albumin as C and then take a log of it and do log of 1 by C versus log of P, because log of P is the partition coefficient. Okay. Now, I can study what is the trend. So, in this particular study, they found that they see all these dots are each individual compound is represented by these dots, and as we discussed previously by the experiment that I discussed earlier, these are the values that are determined. Okay. Now, if I fit a straight line to this, then I find that I get an equation called as log of 1 by C equals K1 into log P plus K2. Okay. So, this is of the form y equals mx plus c. Now, m is the slope and c is the intercept. Okay. Now, using this equation, I can study two parameters y and x, variables y and x. Okay. Now, if I translate this to the equation written below, c is the efficacy of the molecule, and log of 1 by c on the y axis, it gives me the increasing value here, and similarly log p goes up, as you can see here from left to right, and the higher the log p value, the more hydrophobic the molecule is, right. So, therefore, when I go from left to right, I see that the activity goes up. Okay. So, if I fit this to a curve, I get K1, which is the slope, as 0.75, and K2, which is the intercept, is 2.30. Okay. And there are other parameters that we obtain while we are fitting this curve. One is called the linear regression coefficient, which is R, and the second one S is called the standard deviation, okay, or standard estimate of error, right. Now, let us look at linear regression analysis. Now, if you have a set of data points, what happens is that you can actually draw a set of data points. Now, I can fit this to a straight line, okay, and the best fit here is obtained through a software or using numerical analysis, but you can take any two parameters A and B and fit a straight line to it. Okay. Now, you can also have a situation where you have absolutely no trend, right. If I fit a straight line, I actually do not get a straight line, I get something like this. Each of these points is very far from the straight line. Okay. So, therefore, you can have two contrasting situations where there is no trend. Okay. Keep in mind this is again parameter A and parameter B, and here there is a trend. Okay. Now, the linearity here is obtained, I will explain later, but the linearity can be obtained by looking at the regression coefficient. But if I have a linear relationship here, I can then tell you that if I make a new compound in this direction, then the A can be improved. Okay. So, therefore, this is useful in obtaining trends for activity. Okay. Now, here is a data point, for example, that I have fit to a straight line, and I have got, let us say, hundreds of data points. And so, from this analysis, you can see these two arrows here. These are what are known as outliers, which are not really fitting on the line very well. Okay. But you can see that the majority of the points are actually fitting very nicely. There are a few outliers here as well, and majority of the points are fitting very close to the straight line. Okay. Now, when I get this, I can determine the slope, okay, and from this I can also find out what is the value that I get for a regression coefficient. Okay. Now, if we do this analysis, I can do a best fit and then I can do a regression or correlation coefficient, and the closer the value is to 1. So, R should be ideally 1, okay, but rarely you get the situation where R equals 1. So, you would probably end up getting R square, which is the square of the regression coefficient. It would be typically greater than 0.9 would be considered an excellent correlation, or even greater than 0.8 is acceptable. Okay. So, this is how we go about and obtain what is known as a linear regression coefficient. Okay. And what it means is that if I have a sample set of n, and typically the sample set has to be greater than 20, R square, if it is 1, this is highly unlikely. It in fact almost never occurs that you have a straight line that happens between a biological property and a physicochemical property of a drug. So, for example, inhibitory activity versus hydrophobicity, you would almost never get 1, but there are cases where you get R square of greater than 0.8. Okay. So, let us say the R square is 0.85 in a particular example. That means that 85% of the biological activity is determined by the physicochemical property of the molecule. Okay. So, this helps us understand how the activity of a particular series of compounds can be tuned. Okay. So, just to recap, so if I have an activity A, or in this case 1 by A, and if I have a physicochemical property B, if we see that the trend is like this, and if we fit this to a straight line, okay, we can then determine the linear regression coefficient R square, and if the R square is greater than 0.8, that would be all right for us. And if what it means is that the particular physicochemical property B has a major effect on the activity. Okay. Now, there is another term that comes in which I described earlier, which is standard deviation. So, standard deviation is a concept that we have studied many years back, even in 11th, 12th grade it is being taught, but I will very briefly explain to you what standard deviation is without going into too much detail. I would recommend this as a self study, but standard deviation is basically a measure that is used to quantify the amount of variation. Okay. So, when you have data points which are, let us say, something like this, and you try to fit a straight line to it, for example, right. As you can see, there is a small variation here, whereas there is a huge variation in this case. Okay. So, therefore, even looking at it, I can tell you that the fit is not very good. Okay. But if I want to put a number to it, then I would determine what is known as a standard deviation. Now, therefore, what we do is we typically say that a low standard deviation means that the data points tend to be close to the mean or the expected value, while a high standard deviation indicates that the data are spread out over a wider range of values. Okay. So, in a QSAR analysis, one would like the R square, which is the linear regression coefficient, to be, let us say 0.8 or close to 0.8 or greater than 0.8. And in addition to that, we also do not want a large dispersion, because that makes it difficult to interpret the data. Ideally, the standard deviation should be low in value. So, you would typically find S values that are reported along with the QSAR, and we can also use other statistical tests, which I will not go into detail, but you would want a R square value of greater than 0.8 and an S value to be as low as possible, if you want to make good interpretations from QSAR. Okay. Now, in the example that we looked at, where we looked at serum albumin binding, the interpretation of QSAR is that hydrophobic compounds which have a high logP have better binding to serum albumin. Okay. And it is important because the more hydrophobic the molecule is, the better it binds to serum. If a molecule binds better to serum, then what happens is that it is not interacting with the target. Okay. So, you want the molecule to be interacting with the target and not interacting with the serum, and therefore, you would prefer a molecule which has appropriate logP. Okay. So, despite this, increasing the logP actually improves the activity of the drug in many cases we observe. So, in the case study that we looked at, which is serum albumin binding, we find that as the hydrophobicity of the molecule goes up, the activity of the compound goes up. Okay. So, this is understandable because you have serum binding to hydrophobic molecules, but the problem is that what happens is that if serum is going to bind to your molecule, there is less molecule interacting with its target. Okay. So, this is something that one needs to keep in mind while we are developing and designing new molecules, right. Second thing is that if you improve the logP of the molecule, clearly it improves the activity of the drug, which there are other examples of this, but the drug has to interact with the part of the enzyme. Let us say there is an enzyme, and the enzyme typically, or receptor typically we have studied before, has a hydrophilic exterior and a more hydrophobic interior. So, let us say this is the surface at which it has to react. Now, the drug has to come and interact over here, and therefore, this would be hydrophobic. Therefore, one would expect that the more hydrophobic the surface, the better it is interacting with the enzyme or receptor. Okay. So, does it mean that hydrophobicity is the only thing that is important? Well, theoretically, according to our plot, when you increase the logP value, the biological activity should go on increasing. Okay. But in reality, this does not happen, because what happens is that in an in vivo model, the drug is actually trapped in fat depots. So, there are generous fat depots in people which is used during starvation and so on, and the drug ends up being trapped in these fat depots. Okay. Second thing is that depending on the route of administration, so let us say you want to give an injection to a person, the injections are typically given in aqueous media, and if the molecule is extremely hydrophobic, it is almost impossible to get it into aqueous media, and therefore, increasing logP values beyond a particular point is not desirable. Okay. Now, the other part is that hydrophobic molecules are also more susceptible to metabolism. We have looked at previously ADME, which tells us how to understand metabolism, but the more hydrophobic the molecule, the more susceptible it is to metabolism. So, in reality, what we look at is we actually look at a bell shaped curve, right. So, if you look at log P versus log of 1 by C, so there is a particular point for molecules where the activity is the maximum, and these bell shaped curves are what normally people see, and so you want to look for this range of log P values where you have good range of log P, so that you can also develop the molecule further for in vivo studies. Okay. Now, when we do the QSAR for this series of compounds, you will find that there is a square term in the molecule, which is not unusual because you have a parabolic relationship. Okay. So, but what it means is that when P value is small, then the square of log P is very small and the activity is dominated by the log P value. Okay. Whereas, when P value is large, the square of P value becomes larger and it starts overwhelming the log P term, and therefore, you get a curve like this. Okay. Now, this helps us understand what happens when you change log P over a series of compounds, right. Now, typically most of this is determined by softwares. There are very sophisticated softwares that are available, which can help you with numerically understand and fit it to a particular model and get out good models to understand trends. Okay. Now, when we look at log P and QSAR, there are very few drugs whose activity depends only on log P, although log P is an important parameter, And for example, general anesthetics fits well in the following equation. So, you have typically ethers which fits well in this equation. Log of 1 by C is minus 0.22 into log P the whole square plus 1.04 into log P plus 2.16. So, if I plot this, I would get a parabola like what we have seen previously. Okay. Now, what happens is that because I am able to write out an equation which describes the activity, now because of this I can now find out where I get maximum activity. So, this helps us predict the anesthetic activity given the log P value. This is my x axis. If I have a very low log P, then it is quite likely that the activity would be low. Okay. Whereas conversely, if I have a very high log P, then it is quite likely that I would get a low activity, and therefore, the new set of compounds that I synthesize would have to have this intermediary log P values. Okay. Now, this equation that we derive for this particular set of ethers is not applicable to other classes of antibiotics, but there are typically bell shaped curves that are obtained for other classes as well, but they have different constants in their equation. Okay. So, nearly all classes of anesthetics are affected by hydrophobicity of the structure, and therefore, it is possible to understand for each class of anesthetic what is better at getting into the central nervous system, defined here as CNS, and what we typically find is a log P of around 2 would be highly preferred. Okay. And this kind of a study has been applied to 3 examples I have seen here, where you see halothane, chloroform and ether. The log P values goes from 2.3 to 1.97 to 0.98. Okay. So, what we find in the anesthetic values is this is actually the better compound compared to ether. Therefore, now the next set of compounds if I want to prepare would be in around this range where the value is between 1.5 and 2.5. Okay. Keep in mind that if I go very high, that also creates a problem. But what it also helps us do is that if there is a compound with log P of around 2, one could predict that this molecule will enter the central nervous system and perhaps start acting in the brain or in the spinal cord. Here is an example of something whose problem has been addressed by using a quantitative structure activity relationship. This is an example of a molecule which was considered as a cardiotonic agent, but it was found in many patients to induce visions. Okay? And so, these visions are symptomatic of your drug interacting with the central nervous system. So, now based on the previous study, we know that log P of 2.59 can create trouble and it may help us get into the CNS. Therefore, next set of molecules that they synthesized was with the log P of 1.17. And we will look later about size, but SOCH3 is of similar size to OMe, and therefore, this molecule which has basically a difference in the side chain here was found to have excellent activity, cardiotonic activity, but does not induce visions. So, this is an example of how understanding a good structure activity relationship can help us design better compounds. Now, what we will do is we will look at how to determine what is known as a hydrophobicity constant. Okay. So, let us start with benzene. So, if I want to study what happens when I put a substituent, the example I am taking is chlorobenzene. So, here benzene's log P value is 2.13, whereas chlorobenzene is 2.84. Now, I define a parameter called as pi, which is basically nothing but the difference between the log P of the substituted benzene and benzene. Okay. So, here in this particular case, the pi of Cl would be log P of Cl minus log P of H, right, which is basically 2.84 minus 2.13, and that gives me a value of 0.71. Now, the interpretation from this 0.71 is that let us say I have a complicated structure with many substituents, let us say I have R groups here and so on. Now, if I substitute one of these positions which has a hydrogen with Cl, okay, I can suggest that the log P value may change by around 0.71. Okay. So, therefore, this hydrophobicity constant is very useful to understand how the substituent affects the log P value. Okay. So, here we look at another worked example. The pi of an amide here is CONH2, can be determined by subtracting out the log P of benzamide from the log P of benzene. So, here the value would be 0.64 minus 2.13, and I get a value of minus 1.49. Okay. So, if I introduce a substituent CONH2 substituent on benzene ring, it is likely that the hydrophobicity would go down. That means, the molecule will become more soluble in water. Okay. So, let us look at an example now. Here is a table that I have given below. What is the log P value of metachlorobenzamide? Now, if you want to look at it, the experimental value is 1.51. Now, let us look at what happens when we take the values from what we have determined. Okay. So, log P of benzene. So, benzamide consists of benzene, a chloro group and an amide group. So, what I would do is I start with pi of Cl, pi of CONH2 and then add it to the log P of benzene. So, this is nothing but 2.13 plus 0.71 minus 1.49 and the value comes to 1.35. Okay. So, if I compare these two values, these are close to one another and therefore, I have some predictive value by using pi. Now, a similar table can be constructed for aliphatic compounds, and if I change the substituent on the aliphatic compound, let us say I put a Cl on let us say a long chain alkane. If I put a Cl, then it is going to increase the hydrophobicity by a value of 0.39. Conversely, if I put in an hydroxyl group, it is going to decrease the log P value by 1.16, right. So, therefore, by using these two parameters, one can understand what is the influence or one can predict the influence of these functional groups on the hydrophobicity of the molecule. Now, we have looked at log P values, we have also looked at pi. They are related obviously, but they are very interesting in their use, right. If only substituents are being varied in a QSAR study, pi can be used instead of P, there is no problem. So, let us say I take benzene, I vary from chlorobenzene to bromobenzene to fluorobenzene to benzamide to benzoic acid and so on, then I can use pi instead of P. Okay. P is actually a measure of the overall hydrophobicity of the molecule. Okay. So, we already looked at how to determine log P or P. It is a partition coefficient and therefore, it is a measure of the entire hydrophobicity of the molecule. Whereas, pi gives us locally what is the hydrophobicity. So, if we are looking at a particular site over here, let us say this x over here. Now, what happens if I change x from hydrogen to chlorine to let us say CONH2 and so on? This will help us understand locally what happens to the binding effect or to the interactions that are possible in this area. Okay. So, both of them are very useful, but they have to be used in the appropriate manner.